Hello and welcome to Digital Medboard. Here we go. Recombinant DNA technology is used to produce a specific protein on a large scale. For example, if your aim is to produce insulin, the raw material you need is the insulin gene. In recombinant DNA technology, the first step is to obtain the gene of interest. Once you have the gene, you need to introduce into E. coli. However, human genes cannot directly enter E. coli. These genes require a carrier known as a vector. The next step is to link the gene of interest with the vector. To do this, the vector must be cut using a special enzyme known as restriction endonuclease, also called the DNA scissor enzyme. This enzyme cuts the vector at specific sites. After cutting the vector, the gene of interest fragment forming what is called a recombinant vector. Why is it called a recombinant vector? Because it combines a prokaryotic vector from E. coli with a eukaryotic gene fragment from humans. This process is technically challenging. So, strong DNA ligases are used to join the fragments together. The next challenge is introducing the recombinant vector into E. coli. Since E. coli is a prokaryote with a cell wall, it won't easily allow the large recombinant vector to enter the cell. To overcome this, high voltage electric currents are applied, which create pores in the cell wall. Through these pores, the recombinant vector enters the E. coli cell. This process is called electroporation. Once inside, the vector replicates with E. coli, leading to multiple copies. As the bacteria divide, the gene of interest is also passed on, resulting in large-scale protein production. Although the term recombinant DNA technology sounds complex, most of the process happens on a culture plate. On a culture plate, competition occurs between different colonies. Number 1. Colonies without any vector. Number 2. Colonies with native recombinant vector. Number 3. Colonies with a recombinant vector. It's essential to identify and allow only the colonies with the recombinant vector to grow. Otherwise, unwanted colonies may outgrow them. To distinguish the colonies, marker genes are used. The most commonly used mark gene is Beta-galactosidase, also known as LAC-Z from lacoperon. Once LAC-Z is chosen as the marker, you must use a specific E. coli strain with a mutated LAC-Z gene. This mutated gene produces only the omega subunit of beta-galactosidase, which is inactive on its own. You also choose a plasmid vector that can produce the alpha subunit. The most commonly used plasmid vector is PBr322. When both subunits are present, omega from E. coli and alpha from the vector, they combine to form active beta-galactosidase. This process is called alpha complementation. Remember, if E. coli is takes up to a active non-recombinant vector, both subunits are produced, resulting in an active beta-galactosidase via alpha complementation. A substrate called X-gal, 5-bromo-4-chloro-3-indole beta-D galactoside is added to the culture plate. Active beta-galactosidase cleaves X-gal, producing blue color colony. Hence, blue colonies is active beta-galactosidase, means no recombinant vector. If E. coli takes up a recombinant vector, the insertion of the gene disrupts the alpha subunit gene. As a result, no alpha subunit is produced. So, failure of alpha complementation leads to inactive beta-galactosidase. Therefore, white color colony means recombinant vector present. Let's have short summary of colony color meaning. Blue colony, active beta-galactosidase, means alpha complementation. Native non-recombinant vector, so no recombinant vector, and no gene of interest. Next, white colony, inactive beta-galactosidase, means failure of alpha complementation. Recombinant vector, so gene of interest present. We got a question. White colonies in blue and white technique denote colonies with. White colonies indicates a failure of alpha complementation due to the insertion of the gene of interest disrupts the alpha subunit gene, which confirming the presence of a recombinant vector. Therefore, option C is the correct answer. If you found this video helpful, don't forget to like, share and subscribe to Digital Medboard. Got questions? Drop them in the comments below. See you in the next video. Until then, stay tuned. Choose.